You can see my screen, right?
Yeah.
Okay, sure. Hi, Ethan. Okay, so yeah, so let's start class now. Okay, so today we'll talk about the chapter 3. Yeah, which is the we discuss about the three type of the method which is used by your cell to transport the material across the plasma membrane. So today we discuss about diffusion and the osmosis. Okay, so actually what is meant by diffusion? Okay, diffusion is a movement of the molecule. You can read on their definition first, because exam they will ask you to write down the definition. They will ask you like define the diffusion. So this is your answer. You should write that. What kind of the particle? So in this diffusion, it will be molecule. And then secondly, from high concentrations to low concentrations, or from low concentration to the high concentrations. So in diffusion, it will be the high to low. Okay, it's just like you are just going down the hill, right? Yeah, see, just like this. You are moving from the high to low concentration. Actually, this concentration it refers to the concentration of the particles to which is inside the solutions. So they are moving down. Yeah, they are moving down. So this is the point that you should include in your answer. Okay. So diffusion, you can imagine that. Yeah, maybe you can have a look on this picture. Uh, yeah, I think this.
S- Sorry. I don't think you're sharing the thing.
Oh, sorry, I didn't share. It. Oh, I will share again. My apology. Uh, okay. Okay, can you see? Yes, can see.
Yeah. Thank you.
Okay, okay, okay. Thanks. So diffusions is a molecule moving from high to low. As you can see that I draw this triangle. This triangle it refers to the molecule. They are moving from the high concentrations to very low concentrations. Yeah. So they are moving down the concentration gradient. Uh, the concentration gradient they refers to the how many molecule they are contains in the region. For example, when you look at this picture, when you are looking at this picture, you can see that this is your plasma membrane, right? This is your plasma membrane. Okay. Yeah, plasma membrane. And this is inside of the cell, and this is outside of the cell. So how the molecule able to move from the extracellular fluid, which is they are from the outside of the cell surrounding, they're trying to diffuse to inside of the cytoplasm of the cell. So they are diffused into it by using the diffusion. Okay, because outside they have a very high concentration. You can see that outside they have a highest number of the molecule, so they're able to diffuse by the plasma membranes into inside the cytoplasm. So diffusion is very simple, just as simple as that. Okay, so then we should know about in what conditions we will do diffusions. So in the living cell, see, usually in the living cell, and also in the membrane, which is they are partially permeable membranes, we will have the diffusion happen. Okay, so this term, what is meant by partially permeable membranes? In biology, you will learn about the permeability. So what I mean by permeability? Permeability, you can refer to this picture. Okay, you have a look on this picture. Permeability. This picture. I think I didn't include in your graphic. It's okay. I will include later. So this picture is show about the permeability. Permeability means that what kind of the molecule able to transport across, able to pass through the plasma membrane. Yeah, this is a plasma membrane. This place. So for your information that the molecule they're able to diffuse pass through the plasma membranes. This is the maybe this is the outside. This is the inside the cytoplasm. This is inside cytoplasm. So it depends on the few factor. So one is they depend on the size of the molecule. If the molecule they have a very small size, smaller size, that means that they're just able to pass through the plasma membrane, just like in this way, see. So small molecule able to pass through it compared with the large molecule. If the molecule is very large, that means that they require some assistance of the carrier proteins, so they're able to pass through it. For example, you can see this molecule. See, they are very, very large. So when they are too large in the particle size, they are unable to just squeeze through the phospholipid barrier. So they require the assistance, which is from the carrier protein. So carrier proteins, they will help to transport the molecule, which is very larger in size, to able to pass through the plasma membrane. So this is we call it small molecule. Small molecule able to diffuse directly through the plasma membrane. And then large molecule able to diffuse, able to pass through the plasma membrane with help of carrier proteins. I think you still remember that in the plasma membrane there's a lot of the carrier protein, right? Yeah. So size of molecule will be the considerations. Then another is this second factor. The second factor, it will be the solubility of the molecule itself. The solubility means this one, solubility. Either it is water soluble or lipid soluble. If it is a lipid soluble molecule, they are able to pass through the membrane. Lipid soluble just able to pass through membrane, because that our phospholipid barrier, they contains the lipid naturally. So they able to allow the lipid soluble molecule to pass through it. Okay, just directly pass through the phospholipid barrier. How about the water soluble? If the molecule is water soluble, that means that just like in the waters, okay, or any kind of like glucose, amino acids, so they are the water soluble, we call as polar molecule. Polar. Yeah. Lipid soluble, they usually call as the non-polar molecule. You will learn this in future, especially when you are going to advanced level of biology. Will be non-polar molecule. Polar molecule, they usually classify and become the water soluble. So example, it will be the glucose and amino acid. So those polar molecule, they are unable to directly pass through the membrane because that they are polar. Okay, but your phospholipid barrier, this plasma membrane, they will be non-polar. Plasma membrane, yeah, their characteristic is they are lipid soluble and non-polar. Because of this characteristic, when any polar molecule, they need to pass through the plasma membranes, they are unable to directly just pass through it. They need the help from the carrier protein. So the carrier protein, they will help to transport those water-soluble polar molecule to pass through the plasma membrane. So require the carrier protein. Yeah. So generally, this is a two factor. They will decide how the molecule able to pass through the membrane, and then by using what kind of the method. Yeah. So in the syllabus, in our syllabus, we will learn about the simple diffusion, which is this first one. Diffusion through the plasma membrane. Okay, this is we call as simple diffusion. simple diffusion. Secondly, we learn about the diffusion through the channel. Okay, this is they require the use of the proteins. Okay, we know as a channel protein. This is a channel protein. Channel protein. So their purpose is they help to transport the polar or water-soluble substance, which is unable to pass through the directly into the plasma membranes, just like the simple diffusion. So they require the help from the channel protein. Okay, so facilitated diffusion, we didn't learn much. Okay, we didn't know about this in our syllabus, so I will take across. But just let you know that, okay, facilitated diffusion is also a kind of methods. They help to transport the molecule, which is the polar molecule, pass through the, we call as a carrier protein. Carrier protein, sorry, carrier protein. Okay, and then last, we learn about the active transport. Okay, so let us start with the simple diffusion first, which is the how the molecule able to pass through the membrane directly without any help from the carrier proteins or channel proteins. So let's talk about the simple diffusion. Okay, simple diffusion. Yeah. So this is example of the simple diffusion that happens in the living organism. Okay, please take a star. Okay, because at exam they like to ask this kind of question. In our body and also in the plant, they use a lot of simple diffusions, help to get their nutrients. For example, in the small intestine, okay, our digested food molecules such as the glucose and amino acids, okay, so they will be transport from the lumens, enter into the intestine, okay, so into the bloods, lymphatic, so they will use about the diffusion also. They will use a diffusion. Okay, so about this digestion, you will learn about this project in the chapter 7, which is a human nutrition. Okay. So I want you to note down about this oxygen, carbon dioxide, water, vapor, and the oxygens. So remember that simple diffusions always involve in the transport of the gas. Simple diffusion always involved in the transport of gas. You can write a note down here. Simple diffusion involved in the transport of gas. For example, the most common one will be the oxygen and the carbon dioxide. So those are usually involved in the simple diffusion. Okay. Leaf. Okay, when we talk about the leaf, you remember that leaf they contains the hole under this. You can have a look on this picture. Yeah, this picture. See, this is the leaf. Okay, and the leaf, you still remember that the upper side there will be the upper epidermis, followed by the mesophyll cell, and the mesophyll cell they are comprised of the palisade mesophyll cell, which is the very long and columnar cell closely packed, and also they have the spongy mesophyll cell, which is they have the large intercellular airspace, and followed by the lower epidermis, which is they have the stomata. They have the stomata formed on the lower epidermis. So the stoma or the stomata actually is a kind of hole they found on the lower epidermis. So they are made of this gut cell. Okay, there's a pair of gut cell help to produce the stoma. Okay, this is a pair of gut cell. A pair of gut cell help to produce a stoma. So how the leaf able to do the gaseous exchange like in human? Okay, in human, we will talk about in human. In human, we will, for example, in human, we will inhale oxygens or we will inhale carbon dioxide. We will inhale in oxygen, right? Yeah, we will inhale in oxygens and we will exhale carbon dioxide. Exhale carbon dioxide. In the leaf, they do opposite way. They will inhale carbon dioxide. See, they take in the carbon dioxide. The arrow is inside. Yeah, they take in the carbon dioxide and they will release the oxygens by photosynthesis. Because photosynthesis, they require the carbon dioxide and the water input, right? And then they will try to synthesize the glucose. Yeah, this is the equation for your reference. Yeah, these are equations. Carbon dioxide plus the water. Okay, so they will make the glucose C6H12O6, and then they will release the oxygens. Yeah. Okay, so this is the equations for the photosynthesis. Photosynthesis equations. So please remember to balance equation. Whenever you write this equation in the exam, you just balance with 6. 6 is enough already. Okay, and then the upside, you can write about the sunlight. Then down the arrow, you can write about the chlorophyll. Chlorophyll, yeah, not chloroplast. Chlorophyll. Okay, so this is how the plant able to take in the carbon dioxide, and then they will release the oxygens by the photosynthesis. So this process, you can see that the exchange of gas, they involved in the stomata and also the spongy mesophyll cell, which is they contain a very large air space. Yeah, so this process is by simple diffusions. So why it's called as simple diffusion is because that when the carbon dioxide intake into the leaf through the stomata, actually that the outside environments, they have the higher concentration of the carbon dioxide, and the inside the mesophyll cell, they have the lesser concentration of carbon dioxide. So naturally, the carbon dioxide would diffuse inside from the high concentration from outside into inside the mesophyll cell. Inside the airspace of the mesophyll cell, we have the low concentration of the carbon dioxide. So this is we call by simple diffusion. Yeah. Okay, so it involved in the airspace between the mesophyll cell. Yeah. So we know about the carbon dioxide, and also when talk about the oxygen, oxygen is they will release from the leaf through the stomata. They will release out. Okay, they will give out the oxygens. Also involved in the airspace between the mesophyll cell. So they will provide the oxygens to the mitochondria in all of the cell. I hope you still remember about the mitochondria. Mitochondria is the organelle found in the. You still remember that mitochondria is the organelle found in the eukaryotic cell or prokaryotic cell? Ito Suyang, want to have a guess? Do you remember this organelle? Very important. Yeah. What is the function for this organelle, mitochondria? You want to have a guess? What is the function of mitochondria? Yeah. You still remember this structure? This like a short sausage. Yeah. Mitochondria, which is found in the all eukaryotic cell. Okay, the function is they will help in do what? They will help to mitochondria the function. They will help to. Yes.
Aerobic respiration.
Yes, correct. Yeah. Very very good. You can answer it. Yeah, they actually their function. Yes, you talk about aerobic respirations. So why they need to do the aerobic respiration is because that they need to release energy from by doing the aerobic respiration. So in the we call as the eukaryotic organism like us, we are classified under eukaryotic organism. So we in our cell we do have the mitochondria because every eukaryotic cell they need the energy. I think I said before, right? The cell need the energy to perform their cellular activity, just like human need money to survive, okay, to pay all their debt. So this is why that the mitochondria, they are found in all cells. So they need the oxygen inputs to perform the aerobic respiration. So from the aerobic respiration, they're able to provide the energy to the cell itself. Okay, so oxygen carbon dioxide, they are all happens with the simple diffusion. Then how about the water vapor? You can see that the leaf with the water vapors. So that means that the water vapor, they will be released out from the stomata pores and then to the outside the atmosphere, which is the air outside the stomata. So actually this is a process that your plants, okay, they lose their, the plant lose their water by, we call a process known as the transpiration. Okay, plant lost water through the stomata, okay, by the evaporation. So this is, we know as the transpiration, okay, transpiration. I'm not sure you heard this term before or not. Transpiration is only can be done by plant, not in human. They only exist in the plant. Okay, imagine that in the morning, in a very cold morning, okay, when you walk to the, your backyards, okay, you can see that the leaf, the leaf, this is a leaf, right? Yeah, this is a leaf, and then the lower epidermis of leaf, okay, is something like this. This small hole is referred to the stomata, yeah. So this stomata, they will open, then they will release the water vapor, not water, but water vapor. So this is we call as transpiration. Transpiration is a process that the loss of the water from the plant in form of the water vapor to the outside environment. So especially the transpiration, there will be increase in the transpiration rate in the very hot day. Okay, so it's how the plant lost the water. Usually is by the transpiration. I think we'll learn in detail in the next few chapter. Okay, so we talk about the lung. We as a human, we do inhalation and exhalation in the every second in our life, right? So in our lung, we have this structure we call as the this structure, see, we call as the alveolus, or alveoli. See this balloon-like structure. Okay, you can see that this balloon-like structure, something look like this balloon. Actually, inside this balloon structure, they only contain the empty air only. There's only empty air inside this alveolar structure. Because our lung actually is filled with air. So there's air inside. So the lung inhale the air. So the air which is inside contain the oxygens. They will pass from the trachea, and then they will reach the lungs. Can we imagine that? Okay, from the nose, okay, the air will diffuse to the trachea, and then to the bronchus. Bronchus means that the 支气管, okay, bronchus, then to the alveolus, alveolus. So the alveolus, they will be connected to the blood capillary. See, this is how the alveolus, which is this balloon-like structures, they are very closely associated with the blood capillary. You can see the red color artery and also the blue color vein. So when you look inside, it will be something like this. Okay, this is how the alveoli, they are connected. They are actually very closely connected with the blood capillary. So this show that, okay, I erase it first. This show that in our lung, okay, lung take in the oxygens, and then lung, they will pass the oxygens through the alveolus. So when the blood capillary, which they contain a very high amount of the deoxygenated bloods, just like in this picture. This picture, they show the deoxygenated bloods. Deoxygenated blood. These deoxygenated bloods, they contain a very high concentration of the carbon dioxide. So when this high carbon dioxide, carbon dioxide rich blood, they flow through the alveolus. See, so they will cause the carbon dioxide to diffuse from the blood capillary to the alveolus. So they will be like unload the carbon dioxide to the alveolus. Okay, then after that, the alveoli inside, they contain the oxygen, right? They contain a lot of pure oxygen. So they will release the oxygen to the blood. So at the end, the deoxygenated bloods, they release the carbon dioxide to the alveolus, and then they get the oxygen from the alveolus. So at the end, they will become the oxygenated blood again, which is a blood filled with the oxygen and also the nutrients. They will become the oxygenated blood again, which is they have a very high amount of oxygen. So these oxygenated bloods, they will diffuse to the body cell. Yeah, this is body cell. They refer to the body cell. So the body cell, they will get the oxygen from the blood capillary filled with the oxygenated bloods, okay? And then they will get in the oxygen, right? And then they will release the carbon dioxide. They will release the carbon dioxide. Because they are doing the aerobic respiration. Remember that body cell, they always do the aerobic respiration. So they always require the input of oxygen, and then they will always release the carbon dioxide. So the carbon dioxide rich blood will be travel in form of the vein. See, this is a vein, for example, vein. Then get back into the lung to be convert into the oxygenated blood again. Yeah, so this story. Okay, so this show that the gas diffuse, okay, from the alveolus to the blood capillary, or from the blood capillary to alveolus, both also by the simple diffusion. Yeah, so simple diffusion is a very, very straightforward process. Okay, yeah, so this is example of simple diffusion. You can have a look. Simple diffusion. See, you can see this video, a very short video. It show about the simple diffusion. Can see, yeah. Let me open the video. You can see they put two water, okay? You see they put the color inside. And then once they remove away the paper, what will happen? You see they'll slowly diffuse to the next bottle of water. So all become purple. So this is known as a simple diffusion, just as simple as that, which is transfer, which is movement of the molecule. See, movement of the molecule from high to low concentration down the gradient. That's all. Okay. Okay, so now we talk about the factor that influence the diffusions. Okay, the factor will be like the surface area to the volume ratio. So if you want your diffusion to happen with the faster rate, that means that you should able to increase your surface area. See, if you get the greater surface area, that means you're able to increase the rate of the simple diffusions. Yeah. Okay, so this is why that this is the reason that in our cell, the structure of our cell, sorry. Yeah. This is the reason that the structure of our cell, they always becoming in this structure. See, very long extension. In the root hair cell that you studied before, they have a very long extension, right? On the root hair cell. And also in the intestine epithelial cell that you studied before, they have this structure we call as the microvilli. Microvilli. And also in our intestinal tract, we have a villus. Those are the villus. Okay, the difference between villus and the microvilli is, this is the villus, right? So actually on the top of villus, they have a very small, tiny microvilli, like something like this. Just that they didn't show in this picture, but actually there is a microvilli presence on the surface of the villus. So this is the reason that they want to have a longer, longer extensions, so they can get in more contact with the enzyme or with the substrate. So they're able to increase the rate of the diffusion. Okay, so this highly folded surface of the small intestine increase the surface area. So the factor that affect the rate of diffusion include that if you're able to travel in the shorter, smaller distance, you're able to have a faster rate of diffusion. That's all. Okay. Yeah. So so this is the reason that why the blood capillary and the alveolus, see, the wall is very, very one cell thick. One cell thick, that means that it's very thin. One cell thick means very thin. Just like the thickness of one piece of cell. So how thin it will be? Okay, very thin. One cell thick means that distribution for the cell which are very, very thin. Oh, sorry. So if you have a smaller distance, you're able to increase the rate of the diffusion. Remember that smaller distance, shorter distance, faster of the diffusion happen. And also the temperature. If you have the highest temperature, you're also able to increase the rate of diffusion. Why? Because that the temperature is very closely related to the kinetic energy. So when the temperature increase, kinetic energy also increase. Kinetic energy, kinetic energy, which is they able to increase the frequency of the collisions between the substrate. Yeah. And then the concentration gradients. It refer to this one. This concentration gradient compare with this concentration gradient. Okay, A and B. Okay, tell me that when you try to put a particle at here, okay, tell me that which able to move down in the faster rate. It will be A or B. Which one? Have a guess. Which able to move down in the faster rate? A or B? You can see the gradient. A has a steeper gradient. See, steeper gradient. Adam Zuyang, which one? A or B? They able to allow the particle move down in the faster rate. A move down in the faster rate or B will move down in the faster rate. Which one?
A.
Yes, correct. Why? Because A, they have a much more different concentration. Okay, so they have the faster rate of diffusion. So this is the three factor that will cause the diffusion. Okay, that's all. That's all for the simple diffusion. Okay, so you should know about the applications. Application is very, very important, okay, because it usually will come out in the exam. Application is referred to this one. This one. This table. Okay, this table. Then now we talk about the osmosis. Okay, another interesting topic. Osmosis. You can imagine that osmosis is just everything very similar. I mean very similar with the simple diffusion. Just that osmosis only include in the water molecule only. Only involved in water molecule. Not other molecule. Like the sodium chloride or the glucose. They are not included in that. Diffusions, they include in a lot of the molecule. Okay, they can be included in the, for example, the gas molecule. Yeah. So osmosis only involved in the water molecule only. Okay, not other molecule. So the osmosis, the definition is a diffusion of the water molecule from the dilute solutions to more concentrated solutions across the semi-permeable membrane. Okay, you can write your definition in this way. Osmosis is diffusion of water molecule from high water potential to low water potential. What is mean by high water potential to low water potential? It's okay. I will explain to you later. Then down, see down again, down the concentration gradient. And please add one more words. Across semi-permeable membrane. So when the question asks you define the osmosis, then they will give you 3 marks. So you write down this diffusion of water molecule, okay, from the high water potential to low water potentials across the semi-permeable membrane. You will get a full mark. Okay, so let's understand what is osmosis. Osmosis, okay, you can look at this picture. This picture show a very great idea about the osmosis. You can look at, you see, osmosis, they involve in the two type of solutions. Okay, in the solution A and B, you can see that the yellow color is refer to the solid. The blue color is refer to the water molecule. For the B one, you can see that in the A, they have a highest amount of the water molecule, but lesser amount of the solid. So in the A, there will be the high water potential. Here, high water potential. High water potential, but the lower concentration of the solid. Then how about the region B? Region B, they have a large amount of the solid particle compared to the water molecule. So B, they have more solid compared with the water. See, more solid than water. So B, they know as the low water potential. So always remember that osmosis only involved in the diffusion of the water molecule only, not include other solid molecule. So when the osmosis happens in this beaker A and B, what will happen is the water molecule, they will be diffused from the region with they have a very high water concentrations or very high water potential to the regions which they have a lesser water molecule with a low water potential. So the water will diffuse through the semi-permeable membrane like this. Only water molecule move. So at the end, the result, maybe after 1 hour, okay, you can imagine that after 1 hour, you will see in this level. Means that region A and region B, now region B, they have they have a higher level of the water compared with the region A. Okay, because in the region A, they already diffuse the water molecule to the region B. So now the A and B, they both reach the equilibrium. See, they reach the equilibrium. So this means that the water molecule, they're able to diffuse, okay, from the high water potential to the low water potential. So when you're able to calculate it, for example, initially, okay, A and B, A, you know that they have more solute than the water. Sorry, B, you know that they have more solute particle compared with the water particle, right? More solute than the water. Then the A, you have more water compared to solid. More water to solid. Okay? So sometimes the question they will ask you in this way. In the A and the B, they will give you the molarity. For example, in the A, since that you have more water compared to the solid, so the molarity will be around how many? Maybe about 4 4 molar. See? 4 more, okay? So this molarity, they refer to how many solute inside. Okay? Molarity is referred to solute, yeah? Then how about the B? The B, they have more solute than the water. Okay? More solute than water. So in the B, it will be about 6. 6 more. Okay? There will be a 6 molarity. The 6 molarity means that because they have higher concentration of the solute compared with water, so they have higher molarity because they have more solute, the solid molecule. So since that the A will diffuse the water to the B, so at the end, the A, okay, they lost the water, right? See, they lost the water, the A. The B, they get the water. So when they reach the equilibrium, that means that both molarity, they will reach the same in the region A and B. So the A, they will get about 5 molarity. B, they will also get about the 5 molarity. Why? It's because that since A, they have more water initially, so they get 4 molarity. But however, A, they lost the water already. The water are being diffused to the region B. So since A lost water, they will cause the solid, okay, maybe more than the water molecule. So that's why, that's the reason why the molecule will increase to from the 4 molarity to the 5 molarity due to loss of the water molecule in the region A. Then how about in the region B? In the region B, initially, they have more solute compared to the water. But however, the B, they get more water molecule from the A. So slowly that the water will outweigh the proportion of the solute. So that's why the molarity, the molarity will decrease from the 6 molarity to the 5 molarity. Because the addition of the water. So you can imagine that the water will be dilute the solute. Water, they will dilute the solute. So this will cause the reduction of the molarity from 6 become 5. Yeah, so this is a story. I hope that you can understand. Okay, so the water is also moving down its continuous gradients through the partially permeable membrane. You can write it as partially permeable membrane or semi-permeable membrane. The permeability, this permeable membranes, that means that on the membrane, there's a small hole on it. See, small hole like this, small hole on it. So this hole is not a big hole. It's just a very, very small hole. This hole is just small enough to allow the water pass through it. Yeah, so they call it as a semi-permeable, okay? Only allow the small water molecule to pass through it, but not other larger water molecule like the solid molecule, for example. What kind of molecule unable to pass through this semi-permeable membrane? It will be the, for example, sucrose. Okay, sucrose. Or starch. Sucrose is a kind of, we call it as a disaccharide. It's a disaccharide. It's a kind of complex sugar. Okay, starch is also a very, very complex molecule. It's very large, it's very complex, so they know as polysaccharide. It's a polysaccharide. Polysaccharide. Okay, you learn about these two terms in chapter 5, I think. Yeah, in the chapter 5. So here's the story. Okay. And then when you study about osmosis, you should able to classify the solution from isotonic, hypotonic, and the hypertonic. So actually, what is meant by the hypotonic? Okay, maybe we learn about hypotonic first. Hypotonic, that means that the solution have higher water molecule compared to the solid. Yeah, so hypotonic means that the water molecule content is higher than solid. See, for example, you can compare with the solution A and the B, A and B, right? So the A, they have a very low solute, but they have higher water potential. and the B, they have more solute. So the A, they will be hypotonic. A will be hypotonic. Then B will be hypertonic. Hypertonic is because that they contain much more solute particle. So the water will be they will be diffusing. Okay, they will be diffusing from the solution A to solution B through the semi-permeable membrane. Yeah, they will diffuse in this way. Okay, so after diffusion occur, solution B, there will be higher level than solution A. It will be something like this. Okay, so like this, like this. So they still contains 3 solute inside. Solution A still contain 1 solute inside. Just that the water level already being different. Okay, in solution B, water level will increase due to water diffuse from A to B. Then how about the hypertonic solution? Hypertonic solutions means that the solution itself, they contain more solute compared to the water. So more solute than water. Solute more than water. So when you look about the solution A and B, you know that solution A, you have the higher solute, okay, than solution B. So solution A, you are classified as the hypertonic. Yeah, hypertonic. Solution B, you classify as the hypotonic. Hypo. Why? Because that solution B, they able to diffuse the water molecule from B to A. So the water will be moved in this way. So at the end, the result will be in this way. Okay. Also the same solute, but the water level already being high. This one, they have a very low water level. So this is also A and B. So when you write about the answer, first you should identify that which solution is hypotonic, which solution is hypertonic. Remember that hypotonic is water more than solute. Hypertonic is solute more than water. Then water will be always moving from hypotonic to hypertonic. This is a golden rule in the osmosis, okay? Water will also move from the hypotonic. Maybe I write down hypotonic. Hypotonic, or called as high water potential, or very high water concentration. Water more than solid. Okay, they will diffuse to the hypertonic. Hypertonic. Which is they have a lower water potential. Lower water concentrations. And in the hypertonic, they means solute more than water. Yeah, they always move in this direction. Okay, so this is known as osmosis. Only involved in the water molecule only, but not other solid molecule. That's all. Then how about the isotonic? Okay, have you drink of the isotonic drink before? Like 100 plus? Okay, especially after you doing the exercise, right? After you complete the exercise, you lost a lot of sweating. So you need to drink some isotonic solution to to replace back the fluid that you lost. So isotonic solution, that means that both A and B, they have the same concentration. You can see that this solution A and this solution B, solution A, they have 3 solid. B also has 3 solid, right? So they have the same concentrations. Concentration, they refer to the molarity. For example, this solution A, they have 3 molarity. Solution B, they also have 3 molarity. Yeah, both are the same. Okay. So how about the hypotonic solution? Hypotonic solution, they always have a lower molarity compared to the solution B. This is because that, you see, so for example, this hypotonic solutions, they have about maybe three, no, maybe four. Okay, and this solution B, they have about six molarity. Yeah. So at the end, when you reach the equilibrium, you get about five molarity. Okay, and also five molarity. So both reach the equilibrium. Both region reach the equilibrium. Okay, they they have the same molarity. So how about the hypertonic solution? Hypertonic solution, that means that also hyper will be the six molarity, hypo will be the four molarity. Because molarity refer to how many solute concentration inside the solution. It's not included in water. So if you have a higher amount of solute, you have the higher amount of the molarity or concentration. So at the end of osmosis, you get about 5 mole and also this 5 mole. Okay, so when you reach about same molarity for both regions, is there any movement of water happens? You cannot write that there's no movement of water happens when both solution reach the equilibrium. Okay, there is still movement of water molecule occur after both solutions reach the same molarity, which is the equilibrium. Reach the equilibrium. There is still movement of water molecule, just that there is no net movement of water molecule. No net movement. Water molecule. What is the difference between movement water molecule and also the net movement water molecule? The difference is, for example, you can imagine that this is a solution, right? Okay. For example, you can look up example on these hypotonic solutions. As we see that the water molecule you always diffuse from the hypotonic to the hypertonic. So the arrow will be heading to from the left side to the right side, right? From this side. Yeah. However, there are still some movement of the water molecule from B to A. Just a little movement only. Just a little movement. So majority, the movement will be from solution A to B. Only very less molecule moving from B to A. Just very, very less only. Okay, so this is we call as the net movement. Means that the net movement of the water molecule move from A to B is outweighed compared to the water molecule move from B to A. So whenever both solutions achieve the equilibrium with the same molarity, that means that there is no net movement of water. No net. That means that maybe you have about two water molecule diffuse from the A to B. You still have two water molecule diffuse from B to A. Yeah, so they cancel the effect of each other. So that means that there is no net movement of the water. No net. Net means that they cancel the effect of each water molecule. So far, do you get it, Ethan and Suyang, for the osmosis?
Yes. Yeah.
Okay. I hope you agree. I hope you really get it. Because this osmosis they really like to come out in the exam. Okay. So you can have a look on this hypertonic, isotonic, and the hypotonic. You can see that hypertonic always that very, very high amount of the solute. This is solute. This is solute. Solute more than water. Okay. Isotonic, that means that solute both solute and water are equal. Hypotonic means that the water more than solute. So they always move from the hypotonic to hypertonic. Yeah. So this is the water. See, how they move from the hypotonic to hypertonic, and then this outside hypotonic into the cell which is hypertonic. They will not moving from hypertonic to hypotonic. They will not forever. Okay, so we can study about osmosis theory. Yeah, so osmosis, okay, remember the keyword, water molecule from the high water potential to the region of low water potential. High water potential means that the solution is very dilute. Okay, maybe I can add on here. Dilute. Hypertonic means that more water, right? So it's a dilute solution. Dilute. Hypertonic is referred as the concentrated solution. Concentrated solution. Concentrated solution. So in the hypertonic, they always have a higher molarity. Maybe this is about 8 molarity, for example. Then this is about 4 molarity only. Yeah, that's all. Yeah, you can read about it. Okay, you can have a look on this diagram. See, left side and the right side. Left side has more sugar, less water, so they are concentrated solution. Right side, they have less sugar but more water, so that means they are a dilute solution. So water always move from high water potential to the low water potential, means that they will moving from the region we have, which is more water, to the region we have a less water. So at the end, you will get this level. See, the level will be increased. And this level, they will reduce because they lost the water. Even the water level reduce, however, the molarity, they will be increased. Okay. But for example, yeah, for example, you get about 10. Yeah, this you about 10 molarity. Then this is about 6 molarity only. But by the end, when you reach the equilibrium, they will reach the equilibrium. Equilibrium means that there is no net movement of water. No net movement of water. No net movement of water. So that means that 10 plus 6, 16 divided by 2. So both will read about 8 molarity. 8 molarity also. Okay. Osmosis, they usually, I'm not sure, but I think your school teacher, they will help you to conduct the experiment related to the osmosis, which is they will use the potato strip as a experiment sample. Okay, so maybe I can show you about this potato strip. It's something like this. Okay, it's something like this. For example, this is a potato strip. I should write a better potato strip. Sorry. This is the potato strip. Oh. Okay, so they will put this potato strip into a solution. Maybe they can put it with two type of two type of solution, right? Both also contain the potato strip, just with a different molarity. For example, this A and B. This solution A, they have the highest solute. They have a higher solute. That means that see, this is a solute. Okay. Solution B, they have lesser solute. Just very less. So solution A, they have the highest solute. That means that they put the potato strip in a very highly concentrated solution. Okay, very concentrated solution. Concentrated solution. This B, they will build. They will put the potato strip in the very dilute solution. That means that the water more than the solid, right? So osmosis will happen. So what will happen is if you put the potato strip in the highly concentrated solution with a very high amount of solute, no matter it's what kind of solute, even though it's glucose solutions or the sodium chloride, any kind of solute, so it will cause the outside become hypertonic. Okay, outside become hypertonic. Then potato strip itself become hypotonic. Okay, so Adam and Suyang, you tell me what will happen. What will happen in the next step? Water will diffuse out from the potato strip to the surrounding, or the water will diffuse into the potato strip. What will happen? Potato strip will lose the water or they will gain the water. In the solution A, what will happen? Want to have a try? Or you are thinking? Are you okay? Ethan, Suyang? Am I connected? Hello? Yeah. I think you're looking at the screen, right? Okay. Maybe you're thinking. Do you understand my question or not? Adam Suyang, or you are not understand my question? I mean that you put the potato strip into the solutions with a very high solute concentration. For example, the solute in this solution, there will be the glucose, for example. There's a lot of glucose inside the solutions. So the potato strips, you think it will be losing the water or it will gain water? The answer is, since the potato strips are being put into the high concentrated solutions with a lot of glucose molecule inside it, so the potato strip itself, it will become hypotonic. It will become hypotonic. So remember that osmosis will always cause the water diffusion from hypotonic to the hypertonic. So potato strip, they will be losing water. So water will be diffused out. See, diffuse out. Because water always diffuse from the hypertonic to the hypertonic. So at the end, the potato strip will be in this way. Okay. Maybe after 1 hour, the potato strip will be looking in this way. There will be shrinking like this. Yeah. Okay. So this is we know as the flaccid. Okay, flaccid. Because potato strip lost water. Potato strip, they lost water. They are losing of water. So inside the potato strip, they will be in the lesser water contents. If you want to weight the potato strip, if you want to weight it, okay, the experiment they will design like this. They will ask you to weight the potato strip before and after. Okay, for example, if you weight the potato strip before it will be about maybe 10 gram. So after the potato strip lose the water, when you're trying to weight the potato strip, it will be around 6 gram. So about 4 gram of water is being lost. 4 gram of water being lost to the external environment. Okay, so this is the one of the experiment that you can do with the potato strip to study about the concept of the osmosis. Yeah, so osmosis experiments. Yeah, so you can see that when you want to place the potato strips in the distilled water or sucrose solution, then you try to compare between the change in the mass of the potato strip. So the potato cylinder in the distilled water will be increased in the mass the most. Why? Because that this is referred to this concept. If you put the potato strip into the dilute solutions, means that they have more water. more water compared to solid. So that means that the outside solution, they will become hypotonic. Okay? Outside this solution, they will become hypotonic. This one. Hypotonic. Yeah. Then the potato strip itself, they will become hypertonic. Okay? They will become hypertonic. This one. Hypertonic. Okay? Outside hypertonic, potato strip itself become hypertonic. So the water will be diffused from the hypotonic to the hypertonic. So this means that potato strip, there will be absorbed water. Yeah, due to the diffusions. Okay, so maybe answer you can write in this way. Water diffused from dilute solution with the higher water potential into potato strip with a lower water potential. Yeah, by osmosis. This is the answer that you should write in the exam. Okay, so potato strip absorb the water, right? Potato strip absorb water, and they will gain weight. They will increase in mass. Increase in the mass. So maybe initially the potato strip is 10 gram. Okay, 10 gram. They will become about 20 gram. Just for example. So they are additionally 2 gram come from the water mass. Okay. See, so when you put the potato strips inside the distilled water, they are always with the hypotonic solution, because inside distilled water there is no presence of the solid solution. There's no presence of the solid particle. So distilled water is a hypotonic. So it will cause the water diffuse inside the potato strip, so there will be increase in the mass. Okay, if you put the potato strip in the stronger sucrose concentration, that means that it's a very hypertonic solution. Hypertonic or the concentrated solution. Hypertonic solution or a highly concentrated solution. So this will cause the potato strips to losing the water. They'll be losing the water. So when they lose the water, they will reduce in the mass for the potato strip. Yeah. So this is a kind of experiment that you can do in your school. Okay. So that's it. Yeah, that's it. Okay. You can read on yourself. And then let's talk about the osmosis of tissue, osmosis in the tissue. Okay. Osmosis can be studied by using the plant tissue and also the animal tissue. See, animal tissue and plant tissue. For the plant tissue, we can describe the plant tissue, the condition of plant tissue by using these three kind of words, which is the plasmolyzed, flaccid, and turgid. This one. Okay. Plasmolyzed, flaccid, and turgid. Oh. So in the plant tissue, okay, remember that the plant tissue, they contains the cell wall, right? Yeah, you can see that. This is a plant tissue. Let me draw a plant tissue to you. This is a cell wall. They contain a very, oh my god, why I draw in this way? Sorry. Plant tissue, they have the cell wall. Okay, remember that plant tissue, the plant cell is always rectangular in shape. This is because that they have the rigid cell wall. Okay, they have the rigid cell wall. Like this. A very rigid cell wall. Okay, and then with the very large vacuole at here, and a nucleus, a big nucleus, and a cytoplasm. Cytoplasm at here. So this is a plant cell. This is a vacuole. Okay, this is a vacuole. Yeah, and then this is the cell wall. This is cell wall. And then this is cell membrane. Okay, whenever the plant cell, they absorb the water, okay, means that when you put the plant cell inside the hypotonic solutions, they will be absorb water, right? So the water will diffuse inside. So water diffuse inside the plant cell, actually, they will be intake into the vacuum. They will be intake into the vacuum. So they will be appear in this way. You can see. Once the vacuum, they absorb a lot of water, they will become this way. They will enlarge. Wow, you see, become very big vacuum. They become very big until that is so big, they will press the nucleus to the periphery area. See? And also that the vacuum is so fat until the cell wall will be pressing out. Maybe I can write in this way. The cell wall, okay, the cell wall, they will be slightly bending out. Yeah, like this. Slightly bending out of the cell wall. Okay. This one. So this is a vacuum. You can see the vacuum appear increase in their size. So this is how the plant cell able to absorb the water. Plant cell absorb water, and they will store water inside the vacuum. Remember this. Plant cell will always store the water inside the vacuum. So the vacuum will increase in size. Increase in size, right? See? And become large, centralized central vacuum. So once the vacuum increase in size, they will press on against the cell membrane. Then the cell membrane will exert pressure on against the cell wall. So this is the region that the cell wall will be slightly bending out. Cell wall slightly bend out in the shape. Yeah. So this is the distribution you can write that when you see the plant cell in this way. So this plant cell, they will give a name. They call as the turgid plant cell. This plant cell, they call as turgid plant cell. Turgid plant cell. Because it's so much, a lot of tubular pressure inside the plant cell. See, when the cell membrane they exert the pressure against the cell wall, they will create a tubular pressure. We call it as a turgidity. Okay. Actually, what is meant by tubular pressure? This tubular pressure inside the plant cell. Okay. Remember that plant cell is not like human. In human or in animal, we have a backbone to support our cell. But in plant cell, they don't have the backbone. So how the plant cell able to support themselves to stand upright is they depend on this tubular pressure. So this tubular pressure, they will support on the plant cell to stand upright. Okay, so the plants will be appear in the standing position due to the tubular pressure. Okay, so this turgid plant cell. And plant cell also able to losing the water. Okay, so this is they absorb the water. They absorb the water and then they will appear in this way, which is they will become turgid plant cell. So what happens if they lose the water? Okay, they lost the water. They lost the water when you place the plant cell inside in the, for example, you place a plant cell in the hypertonic solution, very highly concentrated solutions, in the concentrated solution. So the plant cell will lose the water. Plant cell, they will be losing the water. Okay? So let's draw about the plant cell after they lost the water. Also rectangular, no much change. Also outside contain the cell wall, no much change. But they become like this. See? And then this is the nucleus. This will be the vacuole. So what happened to this cell? Okay, this is still the cell wall. Cell wall are still at here. Cell wall. However, this is the cell membrane. You can see cell membrane they are appear in this way. Here, this is cell membrane. This is cell membrane. Cell membrane. Oh, sorry. Cell membrane. This is a cell membrane. Cell membrane. And then the cell membrane. So this cell membranes, this is actually a cell membrane. Can you observe that cell membrane actually they pull away from the cell wall? So you can write it in this way, okay? Plant cell lose water. Right. So vacuole, see, vacuole compared with the original site, see, they already have the significantly reduced in the site of the vacuole, right? From such a big site becomes such a very, very small site. Okay, they reduce in the size. Vacuole shrink and reduce in size. And also the cell membrane pull away from cell wall. You can see, right? Cell membrane pull away from cell wall. So there is a space exists between the cell membrane and cell wall. Space they refer to here. This is a space. See, this is space here. The space. Space exists between cell membrane and cell wall. Okay, so this cell they become flaccid. They lose their turgidity. Okay, they lose the turgidity and become flaccid. So this is a flaccid cell. This is a flaccid cell. This one. It is a flaccid FLACCID, or sometimes we call as is a plasmolyzed plant cell. Okay, so that's a story about it. Then please refer to the picture above. Okay, let me add on. So absorb water, okay, in the dilute solution. So the plant cell will absorb water, then will become turgid. So this is when you put the plant cell in the dilute solution. So this is what will happen. So actually, what is the importance for the turgidity for the plant cell is plant cell they require the tubular pressure to support themselves to stand upright. So if the plant cell they have a lot of water absorbed by the vacuole, so the vacuole will expand, increase in size, so become turgid cell. So you can see the plant cell stand upright. It's very fresh and it stand upright. Stand upright. Then how about the flaccid plant? Flaccid plants, see, they losing their water. When they lose the water, you can see the vacuum reduce in the size and the cell membrane pull away from the cell wall. So there's a space. This white color, that means a space. The space exists between the cell membrane and also the cell wall. So the plant will be wilting. So this will cause a wilting of the plant. Wilting, sorry. Wilting of the plant. Because they lose their water. Okay, then for your information, how the plant cell able to get the water is from, they will mostly absorb the water from the soil water. Because the plant roots, okay, plant roots, they are immersed into the soil, right? And then in the soil, they have a lot of water. So the plant roots, they contain a lot of root hair cell, okay? So remember, the root hair cell, we have very long extensions. So maybe I zoom out the picture. Oh, wait. Yeah, so this is the root hair cell. Okay, then this is soil. So the soil contains a very high water potential. High water potential inside the soil. Soil that have more water potential, more water, also soil that have more water compared to salt. More water than salt. Then how about in the root hair cell, okay? Root hair cell, they do contain the cell set. We call as the cell set of the root hair cell. This one. This place, this place, this place. Okay? We call as a cell set of the root hair cell. Cell set. Cell set of root hair cell, they contains lower water potential. See? They have lower water potential. So that means that the salt is higher than water. The concentration of salt will be higher than water in the cell set of the root hair cell. So this is why, this is how the roots able to absorb the water through osmosis. Because there's a high water potential inside the soil. But there's a low water potential inside the root hair cell, the cell set root hair cell. So the water able to diffuse into it by osmosis. Yes. Osmosis. So this is how the roots able to absorb the water. So they will transport the water to the root cortex cell, then to the xylem vessel. So the xylem vessel, they will act as a continuous column. So they will try to transport the water from the stem to the from the roots to the stem, then to the leaf. Sorry. Oh, let me. Okay. Okay. See, soil water and the soil water root hair cell, they have a lower water potential than the soil water. So the water able to diffuse across the membrane of the root hair cell into the roots by osmosis. So the water will move across the roots from cell to cell by osmosis until they will reach the xylem. Okay, they will reach the xylem. So once they reach the xylem, they will be transported away from the roots by the transpirational stream. Okay, to help maintain the gradients. Okay, what is meant by transpirational stream? You can imagine in this way. Transpiration stream is when the water they reach the xylem vessel. Okay, for your information, xylem vessel are discovered in the roots, stem, and also leaf. So it will be something like this. Yeah, this is a leaf, right? This is roots. Okay, to the stem and then to the leaf. This is a leaf. Leaf. Okay, stem, leaf. Sorry. So for your information, xylem vessel are found in the roots. Then they will be extending outwards, okay, to the stem. then to the leaf. So when the water they transport by the xylem vessel from the roots to the stem, then to the leaf, they will be in this direction against the gravitational force because they are travel upward, right? So, and then when the water travel from stem to the leaf, and then they will lose water from the leaf. Okay, they will lose their water from the leaf by transpiration. Okay, transpiration. So once they lost the water from the leaf, there will be a space, right? There will be a space empty for water. So in order to fill the space for the water, they will create a transpiration stream from the roots to the stem. Yeah, in order to replace water lost from the leaf. Okay, so transpirational stream okay, is to replace water loss from the leaf by transpiration. So the plant lose their water through the evaporations from the stomata pores during the transpiration. It's just like in human, okay? In human, we also lose our lose our water. We lose water in form of urine. Maybe we are sweating. Okay, so we need to replace the water. So the method that we use to replace water is we will drink the water. Okay, we drink the water. But in the plant, they are unable to drink the water. So how they replace the water? They just continuously suck in the water from the root hair cell. So this is a method of how they replace the water losing from other part of the plants. So they will continually absorb the water through the root hair cell. So the water will be travel from the root hair cell until they reach the root contact cell to the xylem vessel, from the roots to the stem to the leaf. Always in this direction. Okay, so that's all. And then, yeah. So remember about the term, how you describe the turgid plant, flaccid plant, and also the plasmolized plants based on the type of solution. Okay, so I think, yeah. So the effect of the hypotonic solutions on the plant cell is, okay, please remember that the distilled water of the pure water, they are always hypotonic to the cell set or the plant cell. Yeah, they're also hypotonic. So they will cause the water diffuse, okay, diffuse from the distilled water into the vacuole. See, they will enter into the vacuole of the plant cell by osmosis. So the plant cell will become turgid. The turgid plant cell, that means that inside they contain a lot of water and they have a very high sugar pressure. So this will prevent more water entering by osmosis. Okay, one thing you need to aware of it is plant cell, they get the water. However, plant cell, they will not burst, they will not rupture. Okay, even though there will be excessive amount of water enter into plant cell, but they will still not rupture. Okay, plant cell will not burst. Plant cell will not burst if there is excessive amount of water enter. Okay, why they did not burst? Because they are being supported by the very rigid cell wall. They are being supported by the cell wall. And then the cell wall is very hard and rigid. So they're able to resist the sugar pressure, okay? They're able to resist sugar pressure. If in the animal cell, okay, if you have too much water enter, it will cause the animal cell to be bursting. Yeah. So next week we'll talk about it, okay? What happened to the animal cell when you place in these three kind of solutions. So I think this is a class for today. And then I got homework for you. The homework is, it's okay, I will send in the group later. Okay, so do you have any questions so far? Okay, so thank you, Suyan and Ethan. I hope you enjoy the class. So we meet in the next week. Okay, bye. Good night.
Bye. Good. Thank you.
Bye bye.